Thanks for coming. It's really fun to do this. We don't get a chance to do this as faculty very often. I really feel honored to get a chance to share some of the work that I do, and sharing this podium with Steve Mezyk is really fun. We approach things from two sides of the angle. We're interested in environmental chemicals derived from us humans. I am approaching it from the biological impact side, and he is approaching it from providing solutions through chemistry of actually chemicals that were produced by chemists. I had to do that to you. Okay, so my job first is to introduce for both of us the issues. So this is our Southern California bite, and one of the things you should see, you should notice the tremendous humanity here interfacing with the marine environment. This is the way we're going to introduce the subject of water quality. So the central Southern California bite, I've focused in a little more. LA, Orange County, Riverside, all of this. This is the second largest metropolitan area in the United States. Again, interfacing with the marine environment. In LA County, 10 million persons, Orange County, 3, Riverside, 2 million, and up north of us and south of us, more. So 15 million human beings are interacting with the environment. Okay? This is one way we interact with the environment. Okay? And we do it in various ways. I would advise only that approach right there. Okay? But this is meant to make a point. Just alone, that alone, there's 15 million urinators, and I'm not going to go beyond that, okay? We have to handle this every day, and this is going to eventually end up in our marine environment. So we have 3 major places where that alone, and there's many other aspects to water contamination, but these are representing the 3 major wastewater treatment plants in our area. So and they together, imagine that, release nearly 1 billion gallons in one day of treated wastewater. Okay? So I don't know how you think of wastewater treatment plants, but they are the heroes. The city of Los Angeles has to handle 350 million gallons a day, MGD. Los Angeles County, same thing. Orange County, a little less, but a tremendous amount. These are some of the 3 largest wastewater treatment plant facilities in the world. Okay? So what are we adding to this wastewater? What's the food for thought? Pharmaceuticals. Not just putting pills down the toilet, which is not advisable for the environment, but you eat the pills, you don't use it all, it goes out in your urine, you metabolize it. There's a lot of ways that the drugs go back in to the environment. Detergents, pesticides, even caffeine. Seattle has a lot of caffeine in its Puget Sound. That's no joke. Okay? Plasticizer chemicals and flame retardant chemicals, which go into things like baby pajamas and computer terminals. So this is just meant to drive home the point. These are just the top prescribed drugs. And we all may know some of these, right? Blood pressure, lipid, glucose control, pain, anxiety relief, hormones, hormone mimics, others, antibiotics, vitamins, all kinds of things. Okay? This is in Southern California. The effluents, this is a recent study that I've been participating in, and in the seawater, all kinds of chemicals. And the seawater is where you would expect some dilution, but still are able to measure quite a lot of these chemicals. Now you think, well, what about the wastewater treatment plants? Well, they are doing a great job, but the problem is that you can't clear the wastewater of everything. So this is influent that comes into a plant. That's primary treated, where they've removed a lot of the particulate matter. And then this is secondary treatment, where most of the particulate matter is removed. By removing the particulate matter, they can reduce contaminants by up to 95%. And this has been documented many times. So this has been an important advancement in wastewater and the treatment of it over the last 100 years. But the problem is that these things still get into the environment. Even that 5%, believe me, is very significant. So there's more to it, you know. It's runoff. Driving a car, the fuels and the whatever happens with the atmospheric contributions of cars. This is stormwater. This is water coming off of the land after one of our large Pacific storms coming in. And you know, boats as well. So runoff is a big thing. All of this comes into this marine environment here, okay? And again, 15 million people, okay? So, you know, one of the things we face here in California is this incredible interface, and Northern California as well, between, this is a large estuary and marine as well, and the large marine environment in Southern California. Okay, so that's the backdrop to what we're going to talk about today. And so I'm going to start by addressing some of the biological impacts, and then I'll pass the torch over to Steve to talk about some solutions possibly. Okay, so my own title might be Pollution in Our Waters, What Happens to the Fish? And the reason we use fish is they're living there, and they're vertebrates, and they have the same hormones and the same physiological systems we have, and they're good sentinels for us to understand what these impacts are. Okay, another thing I want to say is I'm an endocrinologist, and so I'm going to certainly be interested in this, but you should know these are extremely critical for animals. They regulate everything about you, almost everything. And that keeps you balanced, it helps you adapt, it helps you survive. And when you think about the kinds of things that are regulated by hormones, it's all extremely critical for your health, for animal health. Another concept, endocrine disrupting compounds. There's a lot of chemicals out there. Some are going to interact with endocrine systems. When they do that, think of the importance of endocrine systems. That's a critical situation. Okay? So any way that you can imagine interfering with the ability of a hormone to do its thing would be an endocrine disrupting chemical. I'm going to start with a response to stress. It's a neuroendocrine system. I'm not going to talk about these steps, but you perceive a stressor, it's perceived through the brain, and this is a neuroendocrine axis, so it ends up being hormonal, and it results in the release of a hormone called cortisol, which is a steroid hormone. Cortisol is very important to all of us during stressful times. It allows us to adapt physiologically and behaviorally. It has a lot of different kinds of effects. So this is a critical endocrine system. And you can think in the life of a wild animal how important it is. So, of course, this is a very seminal event in the life of this bunny. But stressors come in all kinds of ways. Okay. So I'm going to start in San Francisco Bay. And I love the city. I'm from the area. But look at that. It's talking about an interface. Look at all that cement and look at this estuary right next to it. Okay, so in some sites that we've looked at, we work with some folks up in the San Francisco Estuary Institute and colleagues at Moss Landing Marine Laboratories, which is a CSU institution. We look at various sites, so I'm just pointing that out. And I want to get to the endocrine system, so I'm going to kind of flash forward because I want to try to cover a couple different things. In any case, what you're looking at here is the response to stress, and it's a careful protocol. But what we're doing is comparing a shiner perch, this animal, and this is another fish, the Pacific staghorn sculpin. We're comparing their ability to respond to stress under identical conditions. And you see, we all have heard of Oakland. It's very industrial. San Pablo Bay has its issues as well. And what you see in both species, and these are different fish, these swim up in the water, these are down on the bottom eating bottom things where there's more contamination. And you see quite a blunted response to in cortisol in response to stress. So this endocrine system is impaired, and it's impairing the animal's ability to respond normally, physiologically, to stress. Okay? This is just a second-year study, but showing that there were a host of sites in San Francisco Bay that showed this phenomenon. Okay? And the question now is that associated with environmental contaminants? So, well, a couple pictures of Oakland Inner Harbor. It's got the big port. It's got all kinds of strange industry that I don't understand. These are our collaborators from Moss Landing Marine Lab, and that's actually Jesus Reyes, who's here today right now. And I'm going to show just 2 slides of some contaminants. Let me tell you, we're measuring these in our state-of-the-art facilities at Cal State Long Beach. It's a great facility. Rich Gossett is back there. He's the director of Hermes. It enables us to do forefront research without question. Also, these measurements were done by my graduate student, Claire Wagoner, who Rich knows well as well. So, so it's all very connected. Anyway, what I'm going to do is just show two slides. These are just DDT metabolites. You see a range in certain sites. This one site has high DDT. And then this site, this is showing PCBs, and this is another kind of chemical. And the point here is in Oakland and San Leandro Bay, we see twice higher levels. Now, what I'm saying is there's a lot of complexity here, but what you can do, if you can measure the contaminants and the phenotype in the animal, you can correlate statistically and see what kind of relationships show up. One of the things we found is that these are chemicals, PAHs, polycyclic aromatic hydrocarbons, that would come from exhaust, from combustion, fuels, oils, all of those things, and they get into the environment. And we're seeing really pretty remarkable, statistically significant correlations between this kind of chemical, these PAHs, and this endocrine disruption of the stress system. This is entirely new kind of information. We had a little bit of relationship with the PCBs, but nothing much. Then we measured a bunch of other things, and they didn't really correlate. So this really begins to point to a certain class of chemicals involved in this that comes from, yes, us humans, our cars, our boats, all that. Okay, so now I'm going to take us down to Southern California. We're going to stay on this particular endocrine system. There's our great university. So in Orange County, there's an outfall here between Huntington and Newport Beach, and they use a far field location. So in Southern California, we're going to focus on flatfish. They live at the bottom, so they're subjected to a higher level of contaminants down there. And the horny-head turbot and the English sole will be too. You can see it's sitting right in there in the sand. These are very visual creatures, as you can see. They are predators, and so they're always looking for the next worm that pops up. So these are some pictures, because I know these are kind of fun, so I'm going to go through them somewhat quickly, but we have to trawl down 200 feet down to get these fish, but that's where the outfall pipes are to the wastewater treatment plants. And then we dump the fish, this is actually the crew from Orange County, into a raceway, and then they're sorted by species. And by the way, Long Beach, Long Beach, Long Beach, Long Beach, all of these are alumni of our university. They're sorted by species, and so these are different species, and these guys are great marine biologists. And Ann Dahlke from the city of Los Angeles is here. She's one of these people. I can't do these identifications at all like these guys can. And finally, we get them, and then we can sample them. And we have guys like Jesus here. He's a great exsanguinator, but it's the same way you get blood from us, you get from a fish. You can use a syringe and tap in, and you can measure hormone levels. You don't have to kill the fish to do this. We do take tissue samples as well, because we need to analyze chemicals and stuff in some of our studies. Dr. Julie Coleman, and that's Dana Cianfredi, that's an undergraduate, Tammy Gonzalez, all Cal State Long Beach. So let's get to the English sole. This is a very nice stress response, okay? And this is at the far field location in Orange County. When we go to the outfall of the wastewater treatment plant, we see the same phenomenon we saw in San Francisco. When we look at that other fish, the horny-head turbot, the one with the big eyes, it shows a terrible response to stress. And at first we thought, is this characteristic of the species? It'd be different from most vertebrates, really. But then we've been able to capture horny-head turbot at other locations outside of more direct impact by us humans. We do see that we find fish that can respond normally. Okay. These are English sole, but in the region, Ventura, Santa Monica Bay, Orange County, always around wastewater treatment plants, you see this phenomenon. This just points to one of the deficits in the animals. Steroids are made from cortisol. Now, you think this is a bad thing, but it's actually a good molecule too. And we get cortisol from it, testosterone, estradiol, all things we need. So cortisol is made by a variety of enzymes in endocrine tissues. Now, it's a very complex pathway, but I'm just going to focus you on two genes. One that helps cholesterol get into the mitochondria, and that's rate limiting. And then we'll go to the last step to make cortisol. Okay, so let's look at these two. And what we see is they are quite reduced. This is Andrew Hamilton's work, and he's sitting here too right now. Quite reduced expression of these genes, showing that they really have an impaired ability to generate the cortisol in the endocrine level. So we're continuing to study this, and in fact we have some graduate students who are coming in who are going to use proteomics to identify what's wrong in these tissues. So we know some of the genes, but now we want to know, and this is another facility we have here at Cal State Long Beach, which is state-of-the-art and very special. It's an opportunity to measure what's happening in the tissue and identify what's happening from scratch, essentially. Okay. So I just wanted to make another point that's really neat. So we have had a recent line of experiments. So this is a kind of a disrupted horny head turbot. So it shows a really poor cortisol response. When we brought them back to the labs in Long Beach and reared them in clean seawater, after 4 weeks, we call it a depuration. Look, you get a return of the response. So it really starting to suggest that there is an environmental factor that we can depurate, get rid of in the animals, and the normal phenotype, the normal thing comes back in the animals. They recover. Okay. So consequences of disruption. So we have this. Cortisol is a very important hormone, and I just want to make a couple of points, and I can only spend so much time with this, I realize. It's related to growth. When you have chronic stress, growth will be impaired. I can't spend time on this today. I did a couple weeks ago. We had a symposium at the college, and I was able to talk a little bit about this. Also impaired defense. And I want to just show a couple slides on this to show a remarkable relationship between the stress disruption, as I call it, and a phenotype. And we're going to go now to another fish called the California scorpion fish. And this work is with Jesus Reyes, he's not on here, but Jeff Armstrong at Orange County and also Julie Coleman. And these animals have parasites that invade their gills, and they suck blood. And if you look really closely, you'll see these little parasites. They're copepods, okay? And then here shows quite an infested gill. It must be terrible for the fish, okay? So this here shows the level of this gill parasite, and it's low in an animal from the reference area, from a cleaner area, and a nice regular cortisol response in the animal. When we go to two sites that are impaired, the outfall and a harbor site near Newport Harbor, and the opposite, you have incredible levels of this parasite and a lack of normal response in cortisol. So we're starting to see some important relationships between endocrine disruption and animal fitness. Okay, so endocrine disruption of this cortisol, they don't respond to stress normally. They will show impaired growth. They show impaired defense and impaired metabolism, which I didn't show. So this is more important than people maybe originally thought. Okay. All right, and they're related to PAHs and possibly PCBs. We also have other things in the environment, and some of them are estrogens, which come from our urine, come from a birth control estrogen called ethinyl estradiol. Dr. Mezyk's going to talk a little bit about that. There's other kinds. This is a plant-derived compound. This is a fertility drug, some pesticides, industrial chemicals. They end up acting like estrogens. So this is a big issue. It's been around for a while. Some fundamental studies on this DES were done in the 60s. We know that these things are having important impacts in animals. So are they in wastewater treatment plant effluents? Yes. So here's normal estrogen, and here's the birth control estrogen, and this is what's coming out of pipes in different places outside of the United States. In the United States, too, this is just a very simple table, okay? So in the Southern California Bight, we're working on it with our colleagues, and yes, it's there, both. Okay. What does elevated estrogens in fish? What does it mean? Well, in males, they produce egg yolk protein. Okay, now think about it. Okay, so they don't make eggs, and they have this. So that's an estrogenized or feminized male. The other thing that you see is ovotestis, so eggs growing in the testis. So you're beginning to feminize a male. These are the kinds of things you see in wild fish. So feminization of male fish, this is a big issue worldwide. We see evidence of it locally. We still have a lot of work to do on this issue, and I want to emphasize that. But let me show you a couple of the wild things we've seen. In the horny-head turbot, we believe because of its particular feeding niche and what it's eating and how it accumulates things, we see in male horny-head turbot as high an estrogen level as females, okay? These are males, these are normal males over here. You're supposed to have low estrogen levels, right? Okay, and so this is pretty wild. It's caught our attention early on. We've done a lot of these studies with the city of LA. This is Ann Dahlke and others, and so I wanted to make sure I pointed that out. These are male English soles. See, they're not impacted, but these horny-head turbot are, and you'll notice in Orange County, it's a more open oceanic environment. Santa Monica Bay is a more closed environment, both impacted by wastewater treatment plants. But we always see this, that the Santa Monica Bay males always have higher estrogen levels, okay? So this would be the wastewater treatment plant in Santa Monica Bay. The OCSD is down here, okay? So it being a more enclosed environment is one of the reasons we believe maybe the animals are more impacted in this region. This is the Hyperion treatment plant going out into the bay. So what are the mechanisms? Well, they may uptake human-derived steroids. That's an easy one. And then I'm going to go to another one. Now, all animals make estrogen, but no animals make birth control synthetically made estrogen. And we see in turbot, we can measure this compound in there. So yes, they are uptaking ethinyl estradiol from the environment, because that's the only place that comes from is from us. And in the San Francisco studies and these other fish, we don't see that there. There's no wastewater treatment plants where we studied. Okay? So it begins to point to, yes, they do. However, there's also this, that they may endogenously, can the testis make estrogen? And unfortunately, or fortunately, I'm not sure for the animal, they do show evidence that they are capable of this. So here is estrogen levels in a male. This is from Orange County, far field, and that's Santa Monica Bay. And see, you can get these tremendous differences sometimes. And so this is very high estrogen levels in the males from Santa Monica Bay. And it's significantly correlated with increased levels of genes. In this case, it's that initial gene. And in this case, it's P450 aromatase. And for those of you who might be not knowing what this is what is the final enzyme to make estradiol from testosterone, as it turns out, not necessarily expected to be in a testis, okay? And we see increased levels, and it correlates with estradiol. So these are very interesting findings that we're beginning to understand, okay? So this is the last set of slides, and then I'm going to move to the end. The high estrogen phenotype, what kind of environmental factors are involved? We're working on this, but here's really provocative, interesting new data that came out. Erica Wansky and Jesus Reyes has helped us, and Claire Wagoner, and I think even Andrew Hamilton. So these are animals taken from Santa Monica Bay with the city of Los Angeles. We brought them back to the lab for that depuration experiment. And look, wow, the males came back to, they went back to being normal males. So these are animals caught in the field, and there's a lot of complexity about this experiment, but animals that came back to the lab after time, they came back to looking like normal males to us in terms of estrogen levels. The genes all showed the same kind of thing. I'm not going to spend a lot of time on there, but these are testicular steroid-producing genes, and notice they all come back down. Now, these do have testosterone, and this is a relatively normal level of testosterone. Is the depuration effect just affecting the whole gonad? Well, not really. Testosterone levels remain at a normal level. So it seems to be an estrogen-specific kind of an impact specifically on estrogen. Now, I wish I could have time for this, and I knew I wouldn't. That's why I put this cassette in, because I at least wanted to tell you that there is such a thing as thyroidal disruption too. And we just published a paper in this, and it's really, really important. But I'm going to have to flip through. Okay. So excuse me for that, but it is tied to environmental chemicals like PCBs in particular, by the way. Okay, so what do we do about this? There's one answer. So can we somehow reverse this? Well, banning DDT really did help the raptor populations to recover. Banning PCBs in the Great Lakes, bird populations are recovering. Banning PCBs have also been associated, it's believed, with reducing effects on frogs. Okay, what can we do? Well, you can identify the compounds, and you need to know, are they acting? Because that's what's important. That's what I mean by endocrine disrupting compound earlier. You can eliminate the use of unnecessary harmful compounds, hopefully develop substitute compounds that are not as bad. Point source regulation has been very, very successful. Try to stop the production of it and the release of it. And better technologies, for example, for breakdown, and that's what Dr. Mezyk is going to be talking about. Okay, so what can we all do? Try not to flush things. Okay, it doesn't just go into space. Okay, it goes into our environment. Do you really need it? I love that because sometimes we get a little overhyper about taking drugs and things, detergents and all. You know, we use them, but use them well. Use them judiciously. Be supportive of research and education. Our efforts are directed right at solving these issues, right here at Long Beach and at many other universities. And science-based assessment in policy is so important because decisions need to be made with some data behind them, okay? All right, we're doing it for them and we're doing it for us. So I have a lot of things to thank, but I'm gonna just flash it up. The Sea Grant has been instrumental in funding us, and Orange County Sanitation and City of Los Angeles has been instrumental as well. And this is Jesus Reyes. This is a new organization. He's the president. He's a Lome of Long Beach, and he's making a difference, and I'm very proud of him. Collaborators, lots. So I can only just flash this up and find your name if you're here, and thank you. Okay, all right, and a great lab. Okay, so it's on to you, Steve.
All right, well, Kevin has basically taken a fair chunk of the intro, so I don't need to go through it again. But basically, my part of this is, we know we have a problem, and we're going to talk about steroids, but we know we have a problem, how do we get them out? So this is going to be chemistry, and this is going to be about safe water, okay? We can argue that Kevin's right, we can ignore the problem, we can eliminate the problem, but telling people not to use steroids or not to use antibiotics has a few limitations, and people are going to use certain steroids, and they're certainly going to use antibiotics. At the end of the day, though, what our interest here is, it's about making water safe, okay? Either safe for release, safe for consumption, safe in general, and we'd like to kind of concentrate on this bottom $4 signs because realistically, we live in one of the best, well, if not the best country in the world when it comes to health standards, to standard of living, to age expectancy. Everything is based on an incredible standard of living. But that standard of living comes with a cost. Kevin alluded to this already, but our water effectively comes with a whole bunch of other stuff. And this is stuff this is natural, Giardia. The rest of this we've kind of added to the water through various ways. Now, you have to understand the scale of this problem. I mean, there's 300 million, 350 million people in the US. So we're going to talk about steroids in this particular part of it. This is ethinyl estradiol. This is the birth control component. There's two parts, two steroids in the average birth control pill. But to try and get a feel for how big this problem is, if you take all the birth control, I have numbers for about 2004. You take all the birth control pills that were sold in the US. You assume half the population are consuming them. You assume that 10% just gets released through the urine, basically, into the environment, and you end up with almost 100 tons of these steroids being released into the waterways across the US. This is a huge impact. You don't need very, very much. Now, the water treatment plants do an amazing job. They really do. But at the end of it all, they don't get it all out. There are very tiny levels left, but it only takes very tiny levels to have biochemical impacts. So what we're talking about here is how do you get these last little bits out of water on 1 billion gallons a day? That's really what it comes down to. Anybody can purify a beaker of stuff. Okay, you can boil it, you can do a lot of other things. How do you do it when you've effectively got a river of stuff going through and you've got to do it all at once? Well, there are many ways to do it, but remember, traditional treatments aren't working. They're getting most of it out and we need them. I'm not advocating getting rid of adding chlorine because realistically we can't do that. But what we can do is look for other ways to try and get the last bits of chemicals out. And what I'm going to talk today is about the chemistry behind this new set of processes. Now, there are various forms of these. They come generically called advanced oxidation processes. Now we're getting into the chemistry. We're talking about oxidizing these contaminants. And the species that's mostly responsible is right here. It's a radical. It's a hydroxyl radical, and it can be generated by a number of different ways. The utilities, the water utilities, will do things like adding ozone. Now this is after your chlorine and after your filtrations and everything else. At the very end, you add ozone, you add hydrogen peroxide and ozone, UV light. These combinations obviously will break up and make this radical, and this radical is extremely reactive. It's in the water, and it will try and destroy whatever it can get a hold of. So the idea is to try and use this concept to try and do that last polishing, that last cleanup of the chemistry that we want to have. We use two different technologies: electron beams, basically passing electricity through the water; gamma radiation, this is the same as sterilizing your medical instruments, okay, just a hardcore part of the X-rays. And this was inspired in two senses. The first is that electron beams, and that's this thing right here, have been used in the past to sterilize, this is sewage water in Miami. Now this is back in the 70s and 80s. This was a pilot test plant, and it works. Worked very well. This is a thin sheet of water, hard to see here, easier to see here. And this is just basically an electron gun, same as your television, okay? It's just blasting the water. It goes back and forth, and as it blasts it, makes these radicals. The radicals clean up the sewage. The water that they dumped out there in Miami was effectively drinking quality at the end. But if you try to come to Orange County and say, hey, I have this great new technology, all you have to do is spend a couple of $1 million and you can do this, you get told, go away. Okay, you've got to understand, this is new technology. The engineers who are responsible for treating this water can't afford to make any mistakes. They're not going to. They want to know it works. You must prove it before they will do anything at large scale, and that's perfectly understandable. From proof concept, there's engineering and there's chemistry. Engineering is solvable. Chemistry is not really understood. So the role that we've been playing here at Cal State Long Beach is understanding the chemistry so we can basically build the computer models that engineers need to try and quantitatively predict the chemistry that'll occur at large scale. We can do it on the bench. We can do it at a couple of 1,000-gallon level. What we can't do it is at the billion-gallon level, okay? You've got to prove it works before we even think about trying it. So from a chemistry viewpoint, if you're trying to build a model, what we're looking at is basically reaction mechanisms and how fast it goes. How fast, what do we make? Because what we don't want to do is make something worse, okay? We want to clean it up, but if we make something worse, we're in real trouble. So the rest of this talk is chemistry. It's about understanding what we make and how fast it goes. And I want you to keep in mind, it's built in support of the engineering that's required to do this purification. And that's really what we're doing here. Now, no university has the facilities to do this, okay? We need machines or we need systems that are large scale. So basically, this university doesn't have it. This university doesn't have it. If any of you are football fans, you may recognize that golden dome. This is the University of Notre Dame. But on that campus is a Department of Energy facility. It's a very nondescript building. It's probably the ugliest building by far on the campus. And in the basement of that building is a bunch of accelerators. It's a user facility. You're allowed to go there. You can take your students there. And if you pick the right time of year, like January, you tend to find the students don't want to go and look around the campus. They would rather sit in the basement and work. It's amazing how that works. All right, what is an accelerator? Well, it's a mimic. It's a lab mimic of what the advanced oxidation process is. If we take a simple model of water, we can think about breaking a bond here. We can think about ionizing it. Okay, I'm dumping electricity into water. The water's going to do something. Okay, this is really simple. If I break bonds, I'm going to get this particular radical. Hey, that's the one I want. I'm going to get this radical. That's useful. And if I ionize water, I'm going to get electrons. Okay? So that's the simple chemistry. And the only reason for using an accelerator, believe it or not, is it's the most quantitative and easiest way to do it. It's also the most cost effective. I mean, effectively, they don't charge me for using the facility. I can take a couple of students up there. We can work for 4 or 5 days. We get all the data we need. We come back, and as Jeff said, we spend a lot of time trying to understand what it really means. Now, this is a little more complicated because we have this wonderful radical that we want, and we have other radicals here. And we understand that the radicals are just energetic species that do the chemistry we want. But will they do what we want is really the question. We can isolate individual radicals. We can use chemistry. This is easy enough. This is laughing gas. And no, you don't use it for that purpose. But if you actually pre-saturate your solutions, you only get this particular radical, and we can look at it in isolation. The system is straightforward. You take your solution with the contaminant in it that you make, you flow it in front of the accelerator. This accelerator irradiates it, and you look at it in real time. Okay? It's, I don't know, if you had $5 million, you could put one here. We don't have $5 million, so we go there. The cool deal is that they've been absolutely wonderful to myself for 20 years since I've been going there with various students. They're very happy to take students to accept students there. And it's a learning experience that you will not get anywhere else in the world. Now, for specific steroids, the first problem you have is that the steroids in the environment, if you saw any of Kevin's slides, are all at nanogram amounts per liter. We can't do chemistry at that. When we can do chemistry, which is at the microgram or milligram per liter, this is our problem. It doesn't dissolve. Gee, what a surprise. All right, so we can't do chemistry right now. We're having problems doing it directly because basically these steroids are not designed to be soluble in water. They're designed to be soluble in lipids. So we have to play tricks, and the tricks is what takes the time to develop and what we actually spend a lot of time working on. But if we dump a bunch of alcohol in there and work backwards, what we can do is look at a molecule like a phenol estradiol, which is EE2, and this is some chemistry, and basically we can look at real time on a microsecond timescale. We can look at the kinetics of how we oxidize this thing using this radical. Now, not worrying about the details, but of course, like all good physical chemists, you must have a good straight line. There is one. Hey, that's what physical chemists do. We just make straight lines, okay? We prove this. We measured a rate constant. This is how fast the reaction happens. And there's 2 parts on this slide. The first is there was one number in the literature which had been estimated. I mean, 10 plus or minus 1 is a good number, but we got a little better than that. We got about 1.5 plus or minus 0.03. But the other thing to note is that this is actually a very fast reaction. This is about as fast as you can get for 2 species in water coming together. What that means is that it will react, and it is feasible then to use this technology. It isn't very effective if you consider everything else there, but it's effective enough that it can be used to actually do remediation of these contaminated waters. This is a phenol estradiol. It's fast. This is progesterone. This is the other half of your birth control pill, and it's about a factor of 10 slower. And that actually starts to become a problem. Okay, if it's not fast enough, all your radicals are going to react with all the other junk in the water. So it isn't going to be great for this, but this is the one we're mostly concerned about, and it is actually going to work. So the utilities were very happy to have that number confirmed. But then, of course, like all good scientists, you answered one question, you need to ask 3 more. Otherwise, where does funding come from, right? Okay, so the question now comes down to, all right, we reacted. We've got a very highly reactive radical. Where is it reacting? Is it reacting here at this triple bond? Is it reacting here at this ring? Or is it reacting here in the steroid backbone? Well, what you try to convince your students is that the literature has value and you really should go and look at it once in a while. And if you go and take a little model compound, if you look at just this part of the molecule here, it looks like that. You know, someone's measured the numbers, they're all over the place, but they're the right ballpark. If you look at this part here, which is way up there, it's much slower. So just crudely, we can argue that all the oxidation chemistry is happening down here. And believe it or not, the medicinal chemists have known that for years. They said that's what happens. Great. Well, what does that mean? Well, you can look at the chemistry of these rings, and what happens is that the radical actually adds to the ring. You could add here, could add here, could add here. Well, now we have a problem, because we stopped and thought about this, did a little bit more hunting. This species is worse than what you started with. It's really bad. It's a mutagenic, reasonably high steroidal activity. So you've taken your steroid, you've oxidized it. From an engineering perspective, you've achieved your goal, you got rid of it. But what you've made is worse than what you started. This is not good. Now, on the other hand, if you're lucky enough to make it here, it's actually not so bad. No one's really sure if it has steroidal activity, and it actually induces cell death in tumors. You've made an anti-cancer agent. That's not, it's not relevant on the large scale, but the problem we're trying to say is you really do need to know what you make. You can't make it worse than what you started with, okay? That's actually a really important consideration. So part of the thing is now we tried to oxidize this, and we may end up making something much worse. Okay, this is not the way to go. Now, the other thing what happens when you use water and you pass electricity through it and you break it up is you make reducing electrons. So maybe instead of oxidizing it, we reduce it. Maybe we can destroy it that way. So we started playing with this and we looked at the kinetics of this. You can isolate this species by simply adding alcohol again, which you have to add, and it takes away these two radicals, so really easy. We can look at the electron directly. Turns out that if you put a single electron in water, it has a huge, beautiful blue absorption. So you can see it in the red, it's fine. You can monitor it directly. As you add more steroid, it gets faster and faster. This is what we teach in Gen Chem. And you can get a beautiful straight line again by doing the appropriate plot. Now, because we actually have to add alcohol, we can do more steroids, and we started looking at these three here as well. But the two I want to concentrate on are right here, the EE2, the phenyl estradiol, and the progesterone. If you remember before, that one was fast, that one was slow. Now we've reversed it. Well, that's interesting. Again, you go through the model compounds, you look at this end of the molecule, you look at this end of the molecule, and what you find now is that all of the reduction is occurring right there, right at that triple bond. Now, why is progesterone so fast? Well, it turns out that instead of an OH here, you've got a ketone, and it turns out that's a fast reaction. Okay, great. I now know where in the molecule it's reduced, so what? Well, the problem is that that's not the really important end of the molecule. That's the important end of the molecule. So while the reduction works, and works quickly, we're not sure that it's actually doing anything for us, which is really annoying. All right, there's also another problem. Water has oxygen. For any of you who have ever had an aquarium, you know you've got to keep oxygen in the water. When you're treating this at large scale, there's a lot of oxygen around, and you can't use reducing radicals because they all react with the oxygen. So recently, and this is brand new stuff, we're starting to look at this species, persulfate. Now, this is something you can buy. It's a standard chemical. You can add it to water, and it's a water cleanup in its own way. But one of the really cool things it does do is it takes this reducing radical and it makes a different oxidizing radical. And we've started looking at how this particular radical will react with the steroids. And sure enough, same conditions. You need alcohol, you need to have your steroid present. You get nice, good straight lines. We can get rate constants. And we've started to realize that this actually may be a better way to do it. the OH for the ethanol estradiol is making something worse. We know the sulfate reacts differently. We don't know quite what we're making yet. We're still looking at that. And part of the deal is that because this project is only about 1.5 years old, when we're still doing this in bits and pieces, we think we have a new way to actually get this stuff out. Now, there's some large-scale problems. They don't like you adding sulfate to water, because at the end of it all, when you treat water, they add lime. Lime is calcium oxide. You have sulfate in the water, it makes gypsum, or calcium sulfate, and your water looks turbid. It's perfectly fine, it just looks cloudy. Nobody likes cloudy water, okay? One of the rules of life. So it's not perfect, but we think we may have a better way to do it. All right, where do we go from here? Well, this just opened up a brand new field. Obviously, we need to look at more numbers for the remaining radicals and the other steroids. There's a lot of steroids out there. We started with 2 simple estrogenic steroids because they're the most important. One thing we have to do and really lock down is the products. What are we making? Now, one of the great opportunities at this place, in theory and soon, we may have an LC mass spec coming here. That's what we all need. This is what we need, is to try and find the actual products that we make, and then we can quantify how much and whether this is feasible. What are we really making is very important. One thing that Kevin alluded to, and I think nobody's really picked up on yet, is that it's not just the steroids, it's not just the chemicals themselves that are going out. We process them, we make metabolites. Those have a huge impact, and they're more relevant than the steroids themselves. And what we're starting to look at is the metabolites that come from all these steroids in this particular case, and certainly all the other compounds we talked about. And most important at the end of the day, it's not just about getting your one chemical and changing it to something else. What you have to do is take the steroidal activity and get rid of it. It's got to stop being a steroid. Now, we could oxidize it 5 or 6 times, and maybe we can break it up. Maybe we can just destroy it to the point where it's no longer a steroid. But when is it enough? That's really the question at large scale that's the most important. 1 radical reaction may not do it. 5 may be great. Okay, well, if that's the only way you can get rid of it, that's the way you've got to go. There are some wonderful students who've worked on this project. Edsel, Katie, Garrett, Kim. Kim's here. These are undergraduates working at this institution. They go with me to Notre Dame. They slave away, and we're talking 16, 18 hours a day for 4 or 5 days straight. Gets them exhausted, doesn't do much for me. Dion is the guy who got us onto the sulfate radical chemistry as a suggestion, so we thank him for that. And lastly, there are some people I wish to thank. This project is not funded per se. Okay, I don't have an NIH grant or anything like that. This is a project that came out of a need. Kaneshita actually made a personal donation to my group, which kicked this project off. He basically gave a couple of $1,000, allowed my students to go, buy the chemicals, do the work. We started the project thanks to his great efforts, so I want to thank him. One of those students was a Beckman Scholar. She was able to do some stuff whilst working on her own project as well. Women in Philanthropy at CSULB has been absolutely amazing to me. They've funded most of my students at one point or another, and even a small amount of money allows the students to travel, allows them to do this chemistry, and one student was on the MARC program, so I wish to thank them there. At the end of it all, I just wanted to point out that we can do some, as Kevin said, we can do some amazing stuff here. We are at the forefront of what we're doing. Nobody else can do this. No one else has done this. We can do it with the wonderful students we have here at Cal State. And I just appreciate this opportunity to thank you listening to me about some of the fun stuff that we've been doing over the last couple of years. So I'm assuming Kevin and I will answer any questions, but thank you very much for listening.